Yes, welcome all to the next session of the MMD course. Very much welcome also on behalf of Luis Obrevilla. I'm Harry van Goor from the University of Groningen in the Netherlands. We have 3 speakers today, exciting speakers, exciting subjects. So I really hope you're gonna enjoy today's session. Please ask your questions again, like last time, ask them in the chat and I will select a couple of questions. I can probably not have the speaker answer all the questions because of their time limitations. There's 50 minutes for the presentation and then 5 minutes of discussion, and we hope to end the session in about 75 or 80 minutes. The first speaker is Carmen Vasquez from the University of Sevilla in Spain. She's a full professor in the University of Sevilla. She worked all over the place, among others in the Department of Physiology in Johns Hopkins. She worked in the University of Barcelona, in the Technical University in München, and also in the Pontificia Universidad Católica de Chile. Her main interest is the mechanism of organ damage in several diseases, among others, Pre-Eclampsia. And the title of her presentation, as you can see on the screen already, is Use of Circulating Anti-Angiogenic Factors to Deepen into the Pathogenic Mechanism of Pre-Eclampsia. Very exciting. So, Professor Vasquez, please go ahead.
Okay, good afternoon. Thank you very much, Harry. Well, first of all, I would like to thank the organizers for inviting me to participate in this international postgraduate course titled Mechanism of Metabolic Disorders. In this course, I am going to talk about the use of circulating anti-angiogenic factors to deepen into the pathogenic mechanism of preeclampsia. Well, I don't know what happened. Now. Well, preeclampsia is a common pregnancy complication affecting 4% to 14% of pregnancies worldwide. It is a multisystem disease exclusive to humans. Also, it is an important cause of both maternal and fetal morbidity and mortality, and it is responsible for more than 500,000 fetal and neonatal deaths and more than 70,000 maternal deaths per year over the world. According to current guidelines by the International Society for the Study of Hypertension in Pregnancy, preeclampsia is defined as one onset of hypertension. I mean hypertension de novo after 20 weeks of gestation. So the systolic blood pressure is bigger than 140 millimeters of mercury, and diastolic blood pressure is bigger than 90 millimeters of mercury, and the coexistence of one or more of the following new onset conditions: proteinuria and maternal organ dysfunction, such as kidney injury, liver dysfunction, neurological events, hemolysis, thrombocytopenia, or uteroplacental dysfunction like a fetal growth restriction. In addition, preeclampsia can be classified in moderate and in severe preeclampsia. In the case that we have severe preeclampsia, it means that the hypertension is higher with a systolic blood pressure of 160 and diastolic blood pressure of 110 millimeters of mercury. In addition, we can classify the preeclampsia in early preeclampsia and late preeclampsia. Early, when the preeclampsia occurs before 34 weeks of gestation, and late, when the preeclampsia occurs after 34 weeks of gestation. In the case of early preeclampsia, it is present with placental ischemia, I mean unimpaired spiral artery remodeling, and it is characterized by fetal growth restriction and adverse maternal and neonatal outcomes. However, late preeclampsia is linked to maternal factors like obesity, diabetes, is characterized by a low rate of fetal involvement, and it has more positive perinatal outcomes. So what is the pathogenesis of preeclampsia? Well, preeclampsia occurs with an abnormal placentation that leads to an impaired spiral arterial remodeling and reduced placental perfunction. This placental dysfunction makes placenta to produce high levels of anti-angiogenic factors, such as SFLT1 and S-endocrine. SFLT1 is soluble like tyrosine kinase 1, SENG is soluble endocrine. So preeclampsia is characterized by high levels of anti-angiogenic factors and low levels of pro-angiogenic factors, such as PIGF and BEGF. PIGF, placental growth factor, BEGF, vascular endothelial growth factor. So, in fact, the ratio SFLT1 PIGF is a useful biomarker for the early detection and prognosis of preeclampsia. Furthermore, we know now that SFLT1 and S-endocrine from placental origin are responsible for the clinical manifestation of this disease. Therefore, we can confirm that preeclampsia is the most severe among hypertensive disorders of pregnancy. In spite of numerous articles have been studied in the last years, the etiologic mechanism, the prevention and the treatment of preeclampsia using different experimental design and using clinical trials, nowadays there are no effective therapies to treat preeclampsia. In this talk, I am going to explain some results about the use of circulating anti-angiogenic factors to deepen into the pathogenic mechanism of preeclampsia. First, I will explain some results about the role of soluble endocrine in the maintenance of preeclampsia. And next, I will explain some results about the effect of excess circulating SFLT1 on placental dysfunction. The first study we studied the role of soluble endocrine in the maintenance of preeclampsia, as I mentioned before. We use endothelial cells, human umbilical vein endothelial cells, and these cells were incubated with soluble endocrine. We carry out two groups. In the first group, control cells incubated with 5 nanograms per milliliter of soluble endocrine, 5 nanograms because this is the normal amount of soluble endocrine in normal pregnancy. And the second group incubated with 40 nanograms per milliliter of soluble endocrine because this is the normal amount of soluble endocrine in preeclamptic women. These cells were incubated 24 hours. After this time, we call it the cell and we determine NADPH oxidase activity, and we carry a study with blot RNA in order to make experiments to determine protein and gene expression of some isoforms of NADPH oxidase, NOx, 1, 2, 4, 5, nitrotyrosine levels, and expression and activation of INOS. INOS is the endothelial nitric oxide synthase that it is an enzyme very important to production of nitric oxide. Here we have the result. We can see that the cells incubated with high level of soluble endocrine presenting high activity or higher activity in the NADPH oxidase when compared with the cells incubated with low levels of soluble endocrine. Here we can see the expression, protein and gene expression of the different isoform of NADPH oxidase, and we found an increase in the protein and gene expression in NOx1, NOx2, NOx4, and NOx5 in the cells incubated with high levels of soluble endoglin when compared with control. In addition, we measured the levels of nitrotyrosine protein and the gene expression of inus and activation, and we found that soluble endoglin increases the levels of protein nitrosylation and reduce protein expression and activation of inus. As you know, both parameters are very important as biomarker of oxidative stress. So all these results suggest or indicate that not only is soluble endoglin involved in the clinical manifestation of preeclampsia, but it is also responsible for the maintenance of preeclampsia. I mean, preeclampsia produces high levels of soluble endoglin that leads an elevation to oxidative stress that results in more increase in soluble endoglin. So a positive feedback mechanism is established to maintain the development of this disease of preeclampsia. In the second study, we worked about the effect of unsex circulating SFLT1 on placental dysfunction. In this case, we use pregnant rats. These pregnant rats were assigned to two groups, control, normal pregnant, and preeclamptic group. This group was carried with animals which were induced early preeclampsia by infusing SFLT1 at a rate of 3.7 microgram per kilogram per day from day 7 of gestation via a minosmotic pump. This is the preeclamptic group. These animals were maintained until gestational day 19, and we measure systolic and diastolic blood pressure on GV5, 11, and 19. In gestational day 18, GV18, we use metabolic cage, and rats were placed in metabolic cage in order to get urine to determine protein. On GV19, animals were sacrificed and fetal and placental weights were determined. Also, in placental, we measured the ratio SFLT1, PIGF, supersite anion production, activity of NADPH oxidase, and gene and protein expression of isoform of NADPH, inos, nitrotyrosine levels, arginase 1 and 2, antioxidant enzymes, and inflammatory markers. Here, I show the animal characteristic in this study. We can see that the systolic blood pressure and diastolic blood pressure are increased in GV11 and GV19 when compared in preeclamptic animals when compared with normal pregnant. In addition, an increase in the protein urinary excretion was observed in preeclamptic animals when compared with normal pregnant. When we measure the fetal and placental weight, we found a decrease in the weight of the fetal weight and a decrease in the placental weight. Later, we measured the levels of SFLT1 and PIGF and the ratio and the level of soluble endoglin in placenta, and we can find that the ratio SFLT1 PLGF is increased in preeclamptic animals when compared with normal pregnant. An increase was also observed in soluble endoglin in preeclamptic animals in comparison with normal pregnant. So we can say that we are produce early preeclampsia in this animal because we have observed an increase in blood pressure, an increase in urinary protein, and an increase in the ratio SFLT1, PLGF, and insoluble endoglin. Here we can see the production of superoxide anion and nitrosamine levels in placenta, and we observe an increase in the production of superoxide anion in the maternal and fetal phase in preeclamptic animals when compared with normal pregnant. This is the image of the preeclamptic animal. This is the image of normal pregnant. In addition, when we measure nitrotyrosine levels, these levels were increased in preeclamptic animals. According to the activity and expression of NADPH oxidase, we found an increase in the activity of NADPH oxidase. And when we measure the source of superoxide anion generation, we observe that when we use in preeclamptic placenta from preeclamptic animals, we use rotenone. Rotenone is an inhibitor of mitochondrial chain of electron transport. This increase in preeclampsia was not inhibited totally. So it means that a big part of the oxidative stress produced in preeclampsia is due to the mitochondria. Or mitochondria has a very important role in the oxidative stress produced in the preeclampsia. Here we can see the protein, a gene expression of NADPH oxidase, isoform 1, 2, and 4. All isoforms were increased the protein expression, the gene expression, and the protein expression. According to nitric oxide metabolism, we found a decrease in the concentration of nitric oxide in placenta from preeclamptic animals, and this result may be due because a increase in the expression gene and protein expression was observed in enzyme arginase 1 and 2. These arginases are involved in the degradation of L-arginine, and L-arginine is very important for the production of nitric oxide. However, an increase in the inos, in the expression of inos, was observed. In addition, we study antioxidant enzymes such as superoxide dismutase, and we observe a decrease in the activity of this enzyme. However, when we measure the expression of the different isoforms of SOD, an increase was observed for SOD1, 2, and 3. The other antioxidant enzymes, such as glutathione peroxidase or glutathione reductase. Glutathione peroxidase, we observe an increase in preeclamptic animals and in the expression of this enzyme. However, in glutathione reductase was decreased, but an increase in the expression was found in the preeclamptic animals, in placenta from preeclamptic animals. Furthermore, we measure the effect of this SFLT1 on pro-anti-inflammatory parameters and PPARs, and a decrease in PPAR was observed in animals with preeclampsia, PPAR alpha and PPAR gamma, and an increase in pro-inflammatory cytokine, interleukin 1, interleukin 6, and interleukin 18 in preeclamptic animals, together to a decrease in anti-inflammatory interleukin 3, interleukin D in preeclamptic animals when compared with normal pregnant rats. So, taken together, all these results indicate that 1, an excess of SFLT1 is sufficient to induce a preeclampsia-like phenotype, since it caused increased blood pressure and SFLT1 PIGF ratio, and decreased fetal and placental weight at 90 GV in pregnant rats. 2, oxidative stress plays an important role in the placental dysfunction found in this model of preeclampsia. Third, we confirm the involved of NADPH oxidase in this oxidative stress, with NOx2 being the main NADPH isoform implicated in this disease, and fourth, the role of superoxide anion from mitochondrial origin seems to be the main source of oxidative stress in this model of preeclampsia. So we can conclude that the use of anti-angiogenic factors, animal models of preeclampsia, and culture cells are essential to identify mechanisms, develop prevention and need therapies, and evaluate safety and efficacy of intervention before evaluation in clinical trials in this disease. Well, finally, I would like to thank my collaborators here in the University in Seville, Professor Alfonso Mate, Doctora Claudia Reyes, Álvaro Santana, and Pablo Espinosa. And thank you very much for your attention.
Thank you very much, Carmen, for the inspiring presentation on the effects of S-FLIT. So please, for the attendees, ask your questions in the chat. They cannot be done orally, so we have to do it in the chat. Carmen, what is I missed the concentration of S-FLIT, what you're using in your model. Is that similar to what we see in humans?
Humans.
The concentration you use in the injection concentration you use in your animals, what is the concentration?
We infusing 3.7 microgram per kilogram per day from day 7. This is the amount very similar that it is in the pregnant women.
Okay, that was basically my
yes.
my question.
And the same was in the soluble endoglin.
Yeah, and since you think that the oxidative stress system is pretty much involved in the initiation of pre-eclampsia, did you do an intervention with the antioxidants in this model?
Well, in this model we have determined the antioxidant enzyme. You can see that, for example, in the case of superoxide dismutase, the activity is decreased, but the expression is increased. However, the other antioxidant enzymes, such as peroxidase and reductase, the result are inverse. The glutathione peroxidase increase and glutathione reductase decrease. So it means that if you have an increase in glutathione peroxidase, you are producing a degradation of peroxide hydrogen and produce water.
Yeah, I realized that, but if suppose you give S-FLIT to your animals and then in combination with an antioxidant, vitamin C or vitamin E, can you rescue the model then?
Well, I don't know. I know that there are several clinical trials using antioxidant, and the preeclampsia is not reversed.
No.
But it's true that now there are some articles using a compound that reverse the oxidative stress from mitochondria.
Yeah.
In that case, the preeclampsia can be reversed. But when you address your compound to the oxidative stress produced by the mitochondria.
We have a question from Louis Grillo. Thank you for a great talk. Did you check if the oxidative and nitrosative stress affects the offspring tissues? Or does this model only lead to local stress? So is there any effect in the offspring tissues in your model?
Well, I don't know. I don't have studied the offspring yet, but I am going to study this in these rats.
Yes, would be interesting. Paolo Valero, thank you very much for your presentation. The effects of of observed in the animal model on oxidative stress, is the effect the same as observed in humans? So the oxidative stress you see in the animals, is that similar to what you see in humans?
Yes, yes. But the only difference is that, well, in this case, you can use rat and you get the tissue from the rat.
Yeah.
It's impossible to, well, you can get the placenta when
Yeah.
the mother finish the pregnant, and the results are very similar. Yes. This is a clear oxidative stress in placenta.
So thank you again, Carmen, for a great presentation. Maybe there are more questions coming up in the chat, so that you can answer them in the chat. Is that okay?
Okay.
So let's see if Enrique Terran, you'll be the next speaker, but you were not present half an hour ago. Are you in now?
Yes, I am here. Thank you.
Fantastic. Fantastic.
Yes.
So I will introduce you, and in the meantime, you can see if you can share your screen. Enrique
Sure.
Terran is from the University of San Francisco de Quito. I hope I pronounced it well, from Ecuador. Very much welcome. He's an MD, PhD. He got his PhD from the College of London in the UK, and he particularly works on different aspects of the pathophysiology of pre-eclampsia. And he did some pioneering work on the pharmacogenetic research in Ecuador. Very much welcome, Enrique. And his presentation title is Pre-Eclampsia and the Impact on Vascular Function. And we can see the screen, Enrique, so go ahead.
Thank you, Harry, and thank you, Luis, for inviting me to participate in this very interesting activity. I will try to summarize in the following minutes what we have been working in this fascinating field of preeclampsia. I'm a pharmacologist, I am not clinician, so most of the activities that we did in a country like Ecuador was to try to move from the lab to the patients, to try to combine basic research with clinical research. And some of our results were encouraging, so we continue trying to get in deep understanding of our findings. Please let me go ahead, starting with something that probably most of you know. There are several reports suggesting that inflammation contributes to the activation or to the dysfunction of endothelial cells during preeclampsia. That was one of the first theories, that preeclampsia might be due to an inflammatory state. So we decided to start very early measuring inflammatory markers during preeclampsia. So one of our first reports show that in fact during preeclampsia there is quite a lot high levels of IL-6 compared to normal pregnancy, and even more than control non-pregnant women. We try to complement these measurements with TNF-alpha, and we found something not as strong as with IL-6. However, the tendency was almost the same. Preeclampsia was related with higher levels of TNF-alpha compared to normal pregnancy and controls. But the interesting thing was when we were to something very more familiar to everyone in clinics and easy to measure, that was C-reactive protein. And now is very well known. As you can see our site, it is from beginning of the 2000. So C-reactive protein maintains the same pattern of IL-6 in preeclampsia compared to normal pregnancy and controls. So at that point, our question was, what is really that meaning? Was CRP and cytokines higher in preeclampsia the consequence of the disease, or that was the reason why the disease was developed. So following that, we decided to do such an interesting study to measure CRP protein during all pregnancy. And we did in women that later on developed preeclampsia and those that maintain as normal pregnant women. And what was interesting is that, as you can see, at the beginning of our measurements, week 16, there was no difference, important difference between pregnant women and preeclamptic women. Probably those with preeclampsia had higher levels, but was not significant. And during pregnancy, the levels were almost the same, and was only at the end of pregnancy that the curves separate. So probably what we were looking with CRP was a consequence of the state at the end and was not something that can predict. And that was our conclusion, that women with preeclampsia had higher levels of CRP at the beginning of pregnancy, but that is not a predictive value for later onset of preeclampsia. So we continue looking for another part of the history, and there was enough evidence that normal pregnancy is related with an increased production or an increased action of nitric oxide. We were part of that initial history. At the same time, there was suggested that during preeclampsia, there might be an alteration, an abnormal bioavailability of nitric oxide that can be responsible for the clinical aspect of the disease. So we decided to try to answer this question, and we set up a very interesting model to measure intraplatelet cyclic GMP levels to try to approach of what is happening. If there is nitric oxide or there isn't nitric oxide. And as we published, was very interesting that normal pregnant women showed higher levels of intraplatelet cyclic GMP compared to control non-pregnant women. However, as you can see, in women with preeclampsia, there were significantly lower levels of intraplatelet cyclic GMP, levels that were a little bit higher than non-pregnant women, but significantly lower than normal pregnant women. That means for us that in preeclampsia, there is not a problem of nitric oxide production. The problem is that that nitric oxide is not working properly. So it was for us a suggestion that for whatever reason, nitric oxide is consumed before it can act and produce their results. So we try to find an answer for our proposal and decide to measure, as we did with CRP, what happened with nitric oxide during pregnancy, normal pregnancy, and in those women with preeclampsia. And we were able to publish this very nice picture. I love this one because you can see that at 16 weeks, there is a huge difference in the levels of nitric oxide between women with normal pregnancy in red and those who later on develop preeclampsia in yellow. And that difference is again reproduced at the end of pregnancy. So we were able to show that if we can measure nitric oxide very early in pregnancy, we can get one of the most sensible detectors of risk for later development of preeclampsia. Our RR was 13. We don't know any other factor that can get that strong prognosis level. Unfortunately, still to measure nitric oxide is not an easy task. It's not something that you can do in a routine laboratory. Therefore, the technological barrier prevents the use of such an interesting tool. Having known that, we were to try to understand what is capturing nitric oxide then. What is the reason why that nitric oxide is not working in women who develop preeclampsia? And we enter in the field of nitrosophiles. Nitrosophiles, as you probably are aware, are considered natural storage for nitric oxide that is released in physiological conditions. Nitrosophiles is the simple reaction between nitric oxide, ROS, and sulfur-containing compounds. So we were back to our model, to the preeclamptic women, and we found this very interesting picture. We found that women with normal pregnancy has not different levels of nitrosophiles of nitroproteins compared to women non-pregnant. However, look at the end, what happened with the preeclamptic women. Women with preeclampsia have significantly higher levels of nitrosophiles. Therefore, we were aligned with our suggestion. Women with preeclampsia produce, in fact, normal levels of nitric oxide. However, that nitric oxide can be sequestrated, can be taken over by nitrosoproteins, by nitrosophiles, and therefore is not acting at the cellular level. Based on that, we did an experimental approach. We take blood from women with preeclampsia, with normal pregnancy, and we decide to do a chemical breakdown of nitrosophiles to see if we can recover nitric oxide. And as you can see in this picture, when we take the samples untreated, when we measure the levels of nitric oxide, women with preeclampsia show significantly lower levels of nitric oxide compared to normal pregnant and controls. However, when we break down artificially using copper, we show that the levels of nitric oxide were almost the same. So we confirm our theory that during preeclampsia, nitrosophiles are responsible for capture nitric oxide. The question remaining is, why is this happening? So we went back to the ROS production, mitochondrial respiratory change. And as you remember, mitochondria is the first one responsible for the production of ROS, particularly superoxide. But we know that when superoxide is present and nitric oxide is increased, we get the production of peroxynitrite, as was mentioned early in the previous conference. So we realize that the electron transport change is depending from complex 1 and from complex 2 to transport the electrons to complex 3 of ubiquinone. Ubiquinone in humans has a change of 10 carbons. Ubiquinone in humans is coenzyme Q10, and that coenzyme Q10 has 2 stages. Therefore, it can be oxidized and it can be reduced, and depending of the number of electrons, they change from one state to the other. So we decide to measure what is happening with this intermediate in women with preeclampsia. And we were the first group to report that during preeclampsia, there is significantly lower levels of coenzyme Q10 compared to normal pregnant women, and at the same level that control women. So we were in the rationale that women with preeclampsia has a problem with the electron transport chains, and that could be the reason why more superoxide is produced, more peroxynitrite and nitrosophiles are sequestering the nitric oxide. So to confirm this, and again, to raise the point, if Q10 levels lower during preeclampsia is a consequence or is the reason why the disease is developed, we decide to set up a clinical trials giving Q10 to pregnant women early in pregnancy and follow up them to see the late onset of preeclampsia. We decide to work with more than 200 pregnant women. We capture them as early as possible during pregnancy, ideally before week 16, and we start giving them either Q10 200 milligrams or the corresponding placebo and follow up them during the whole pregnancy. As you can see, and is obvious, those women receiving Q10 showed higher plasma levels of Q10 compared to those receiving placebo. But the interesting thing is what happened with the outcome. So this is another amazing result. We were able to demonstrate that supplementation of Q10 was able to reduce almost by half the incidence of preeclampsia in our clinical trial. That was a very significant reduction, and that is something interesting to try to explore. It's obviously depending on the population that we use. However, a very similar result has been found in a couple of studies conducted at the US with American population, suggesting that Q10 can be a very interesting strategy to reduce the risk of future preeclampsia development. Unfortunately, Q10 is still expensive and is not available as we would like. Most of the product containing Q10 has small quantities of Q10. They have 10, 20 milligrams, and we were using quite higher amount in our clinical trial. But the result allowed us to show that Q10 was one of the main reasons why preeclampsia can be developed, and that can be the key element in the disruption of nitric oxide action and the further endothelial inflammatory response seen in women with preeclampsia. All these studies conducted during the last latest 20 years has been possible due to the Universidad Central del Ecuador, where we were during a decade, Universidad San Francisco de Quito, where we have been the last decade, CENACYT, that is the National Agency for Science and Technology in Ecuador, and of course, our gratitude to all physicians and women attending at the Hospital Gineco-Obstétrico Isidro Ayora in Ecuador, one of the biggest one, for allow us to get this research conducted. Thank you very much for your attention, and if you have any question, will be my pleasure to try to answer that. Thank you.
Thank you very much, Enrique, for this inspiring presentation. Let's see, Romina Villarado Fernandez, thank you for this presentation. Are there any off-target effects of Q10 supplementation? What about the offspring? And are there any phenotype or changes observed? So the first one, what about the offspring and off-target effects?
Well, that is very interesting, because all experimental studies demonstrate security of Q10 during pregnancy. When we use Q10, it was interesting that we were not able, for example, to find differences in the newborn weight. However, in another set of studies, what we realized was a different neurological development in the newborns of women receiving Q10 during pregnancy compared to the controlled one. No other specific difference that we can mention now.
Hector Ramel, thank you for amazing presentation. What would explain the loss of difference in NO levels in some points of pregnancy between normal pregnant women and women with pre-eclampsia? So it's about the loss of the differences in nitric oxide levels at some of the points during pregnancy.
Thank you for that question, because that is very interesting, because in another set of experiments, we were concentrating in what happened with mitochondria at the placenta level. And it's very interesting because when placenta begin to develop, is a very energy consumption process. Therefore, what we were suggesting is that in women with preeclampsia, the lack of difference in nitric oxide during most of the pregnancy is the responsible of the lack of good placentation process. So the energy required to placenta to develop properly in women with preeclampsia is due to that lack of nitric oxide difference, because women with normal pregnancy consume that nitric oxide, therefore they can afford the energy requirement and get good placentation. That is why you see the difference in nitric oxide at the end again, when energy is not more required for placentation.
Thank you. Luis Obrevilla, does the treatment of Q10 diminish the symptoms and signs of preeclampsia or alleviate those symptoms?
Yes, that's right. The most important effect is on the blood pressure, obviously, but also reduce other manifestations, other clinical manifestations. Is correct.
Paula Valero, the effect of Q10 is interesting, but it only explains the percentage of cases. In the cases where it did not have any effect, it is because the supplementation was very late in pregnancy, or can it be another cause? So that's part of the patients respond and the other part does not. What is the reason of that?
We feel at the beginning that it might be because we started too late, because we start supplementation at week 20 So our initial guess was that probably the early we start, the better will be the result. But at the same time, we probably don't need to provide Q10 during the whole pregnancy, because as I mentioned early, if the point is to facilitate placentation, we probably should concentrate only in the first trimester of pregnancy. So probably another interesting study will be to provide Q10 as early as possible, let's say week 8, 10, and only up to week 20. And with that, probably the result will be same or better inclusively.
Thank you. There are 3 or 4 more interesting questions in the chat, Enrique. Can you answer those in the chat? Because I would like to move on to the next speaker because of time.
Yes, sure. Without problem. I will do.
Thank you very much for this inspiring talk, Enrique.
Thank you.
If you close your presentation, yes. Thank you. So the next speaker is Ronaldo Maren from the Instituto Venezolano de Investigación Científica from Venezuela. He studies the plasma membrane ATPase activities in health and disease, and also oxidative stress in the human placenta. He unraveled also the role of magnesium sulfate as an antioxidant in stabilizer of the plasma membrane. The title of his presentation, as you can see, ATPase activities and oxidative stress during pre-eclampsia. Ronaldo, this floor is yours. Go ahead.
Thank you very much, Harry. And thank you very much for the previous speakers, because they made easier my presentation. As Harry mentioned, I'm going to talk about ATPase activities and oxidative stress during preeclampsia. Preeclampsia is a clinical syndrome of the pregnant women defined by the appearance of hypertension, proteinuria, and edema after 20 weeks of gestation, with an incidence of 3 to 7% in many countries. It is an important cause of morbidity and mortality among mothers and fetuses, and it is one of the leading causes of intrauterine growth restriction. Hello?
Maestro, ¿cómo vas?
Can you hear me?
Gracias, gracias.
Can you hear me?
Yes.
Okay, okay. It is one of the leading causes of intrauterine growth restriction and premature birth. It is diagnosed by the new onset hypertension with values above 140/90, and it is accompanied by one or more other features such as proteinuria. There is a sound coming from somewhere.
Yeah, I think Enrique's microphone is still on. Enrique, can you mute yourself?
Okay, thank you. And the new onset of hypertension is accompanied by one or more other features such as proteinuria, maternal organ dysfunction, hematological involvement, and uteroplacental dysfunction. It is classified, perhaps in the literature, you can see that terms such as mild preeclampsia, severe preeclampsia, and right now the modern classification states that preeclampsia per se is a critical risk for any pregnant woman. So it is classified as preeclampsia and preeclampsia with severe features that includes blood pressure with 160, 110, proteinuria above 5 grams in 24 hours, platelets below 100,000, serum creatinine above 1.1, oliguria below 400 milliliters in 24 hours, indicating an important kidney dysfunction, elevated liver enzymes, pulmonary edema, cephalia, epigastric pain, visual disturbances, and this could be accompanied by the HELP syndrome, which stands for hemolysis, elevated liver enzymes, and low platelets. And if the whole thing is accompanied by seizures, and then we are talking about eclampsia. There are 2 subtypes of preeclampsia, early onset preeclampsia and late onset preeclampsia. The early onset preeclampsia is characterized by the signs, symptoms, and delivery occurs before the 34th week of pregnancy and is present in between 5 to 20% of all preeclampsia cases, and is characterized by specific trophoblast effects, uteroplacental hypoxia reoxygenation, maternal malperfusion, oxidative stress, severe fetal growth restriction, placental apoptosis, necrosis, placental produced debris. And in the case of the late onset preeclampsia, the symptoms and signs and delivery occur after the 34th week of pregnancy and is present in most of around 80% of preeclampsia cases. And it comes with maternal metabolic defects, abnormal placentation early in pregnancy, oxidative stress, apoptotic trophoblast debris, and endothelial dysfunction. The placenta is believed to be the source of the etiology of preeclampsia, although there are probably numerous etiological causes of preeclampsia at the molecular level. All of these lead to oxidative stress later in gestation. It is associated with abnormal trophoblast differentiation and invasion, resulting in altered vascular remodeling of spiral arteries. This in turn leads to reduced placental perfusion and ischemia, a source of oxidative stress. When a plasma membrane is attacked by free radicals, it is possible that several things could occur in this plasma membrane, such as protein splicing, protein-protein cross-linking, disulfide bonds, lipid-protein cross-linking, lipid-lipid cross-linking, oxidative fatty acid residues, the release of malondialdehyde, and amino acid oxidation. This is very important for the membrane proteins, such as the membrane ATPases, like the plasma membrane calcium ATPase and the sodium potassium ATPase, because they are highly dependent on the lipid structure, in particular, the lipid fluidity of the plasma membrane. And if this is broken, then the activity of those ATPases could be also affected. Okay, the free radicals can propagate when the blood cells passes through oxidatively stressed placenta, and they become peroxidized, and they take hydroperoxides to the distal sites, propagating the damage to several tissues of the pregnant women. This is very important. And as you can see in this table, the levels of lipid peroxidation measured as conjugated dyneins and T-bars in red blood cell ghosts, it is higher in preeclamptic women as compared to normotensive. Is significantly higher. And this has an impact on different cells. Interestingly, there are several cells that showed elevated levels of intracellular free calcium concentration with preeclampsia, in particular blood cells and the syncytial trophoblast of the placenta. So if there is a relationship between the elevated levels of intracellular free calcium concentration and the lipid peroxidation, make us to evaluate the different mechanisms responsible for the fine control of the intracellular calcium concentration. In this cartoon, we can see that the intracellular calcium concentration is controlled by the plasma membrane calcium ATPase, the sodium calcium exchange that uses the sodium gradient created by the sodium potassium ATPase to extrude calcium outside the cell. And also the intracellular calcium could be controlled by the SERCA, which is the sarcoendoplasmic reticulum calcium ATPase that takes calcium from the cytosol to the sarcoplasmic reticulum, and also the mitochondrium can take calcium. But the fine control of the intracellular calcium concentration is performed by the PMCA, or plasma membrane calcium ATPase. So if the source of oxidative stress is the placenta, let's take a brief look of the placenta. Placenta is a fundamental organ during pregnancy. It is provisional and it is composed of embryonic and maternal tissue. It has the appearance of a disc with the umbilical cord inserted in the central part of one of its faces, the fetal face. In this cartoon, we can see the main maternal fetal exchange mechanisms in the syncytial trophoblast. On the left side, we have the placenta, and as you can see, the chorionic villus, which are made of the fetal capillaries, they are immersed in the maternal blood, and the capillaries are separated by two layers of cells, the cytotrophoblast and the syncytial trophoblast. At the term placenta, basically the separation of the fetal blood and the maternal blood is done by the syncytial trophoblast. So there, the control of the maternal fetal exchange is performed by the syncytial trophoblast, and the main transfer mechanisms related to maternal fetal exchange are the endocytosis, exocytosis through membrane vesicles from the basal membrane to the microvillus membrane and vice versa, diffusion channels, mediated transport, active transport. We have shown that the plasma membrane calcium ATPase is present in both membranes, BM and MVM, and it is also known that also the sodium potassium ATPase is present in both membranes. Therefore, we measure in both membranes T-bars as an indication of lipid peroxidation in uncomplicated pregnancies, preeclamptic, and as you can see here, in both membranes, BM and MVM, the level of T-bars are higher in preeclamptic membranes as compared to uncomplicated pregnancy. This increased in lipid peroxidation produces an important diminution of the PMCA activity. As you can see in this figure, there is an important reduction of the PMCA activity. And this reduction in PMCA activity is not due to a diminution or modification of the number of PMCA molecules, since the relative density of PMCA is similar for both membrane preparations. In other words, what is happening is a reduction of the turnover rate of the enzyme with preeclampsia. Similar results were found for the sodium potassium ATPase activity, a reduction with preeclampsia without changing the number of ATPase molecules, indicating that preeclampsia is reducing the turnover rate of the enzyme. In order to evaluate if what is happening with the ATPase activities is due to the oxidative stress or the lipid peroxidation, we performed several studies with placental explants. Basically, we prepare the placental explants from normotensive pregnant women, and we incubate them under control with normoxia at 8% oxygen or hypoxia 2% oxygen. And then we isolated the basal membranes and microvillus membranes, and we measure in them T-bars and ATPase assays. As you can see in this table, the level of lipid peroxidation measured as thiobarbituric acid reactive substances or T-bars is higher for both membranes when the explants were incubated under hypoxia condition. If the explants were incubated under hypoxia, but in the presence of an antioxidant such as BHT, butylated hydroxytoluene, the increase in lipid peroxidation is not seen. This produces an important diminution of PMCA activity with hypoxia, and sodium potassium ATPase activity with hypoxia. There is a close relationship between the level of lipid peroxidation and the ATPase activity. Finally, I would like to mention something about the clinical management of preeclampsia. Basically, what is used is the antihypertensive drug therapy and the use of magnesium sulfate. Magnesium sulfate has several benefits for the pregnant women with preeclampsia. I would like to mention that increases brain blood flux, increases the synthesis of prostacyclins, protects against damage from free radicals. This is very important because up to now, there are no reports that magnesium sulfate could act as an antioxidant. In the following experiments, we used the following protocol in order to study the effect of magnesium sulfate therapy. We took blood samples from normotensive pregnant women and from preeclamptic pregnant women without any treatment. And after 24 hours of magnesium sulfate treatment, and then we isolated the red blood cells, we prepared red blood cell ghost, and we measure in those ghosts ATPase activity and lipid peroxidation. In this table, we can see that the magnesium sulfate therapy reduces significantly the level of T-bars, and in those ghosts, the levels of T-bars are similar to those, not only T-bars and conjugated ions are similar to the ones seen from the normotensive pregnant women. And importantly, the calcium ATPase activity was restored to values also similar to normotensive pregnant women. So in other words, the magnesium sulfate therapy is acting as an antioxidant, and it's very important because there are no explanations for this effect of magnesium sulfate. We also measured the calcium ATPase activity, the transport activity of the calcium ATPase by just preparing inside-out vesicles from this ghost. And as you can see in this figure, the active calcium uptake as a function of time is, there is a reduction of the transport, the active calcium transport with preeclampsia in the absence of the treatment with magnesium sulfate, and with the treatment with magnesium sulfate, the transport capacity of the PMCA is similar, reaches values similar to those from the normotensive pregnant women. In other words, magnesium sulfate could be interacting with free radicals. One possibility could be that magnesium is replacing iron, which is essential for the release of hydroxyl radical from the hydroperoxide, but this is very unlikely because magnesium sulfate does not dissociate so easily. And the other possibility is that could be that magnesium sulfate could be interacting in some way with hydroxyl radical or the superoxide anion. Well, this was tested by a collaboration that we have with the group of computational chemistry with Doctors Ruete and Fernandez in the Center of Chemistry in our institute, and they measured by just performing quantum chemistry calculation with a density functional theory that, and they conclude that magnesium sulfate can react with hydroxyl radicals by preventing the oxidation of cell membranes by stabilization of the radical spin density. Here we have the magnesium sulfate, the hydroxyl radical, and here with the interaction of the magnesium sulfate, there is a stabilization of the free radical spin density. This is very important because this could be the first explanation of why magnesium sulfate can act as an antioxidant during preeclampsia. In conclusion, there is an increased lipid peroxidation of the syncytial trophoblast plasma membranes during preeclampsia with a concomitant diminution of both PMCA and sodium potassium ATPase activities of these membranes. These effects seem to be due to hypoxia of the preeclamptic placenta, and both parameters, calcium ATPase activity and level of lipid peroxidation of red blood cells ghost, return to normal values either after several weeks of delivery or after 24 hours of magnesium sulfate therapy. The magnesium sulfate effect can be mimicked in vivo, in vitro, and it seems to be due to direct interaction with the cell membranes. Magnesium sulfate acting as an antioxidant. Thank you very much. Here I have some recommended readings for the students that they can review, and if they have questions, they can write to me, and I will be happy to answer all their doubts and questions. Thank you very much.
Thank you, Ronaldo, for a beautiful presentation. For the attendees, please again ask your questions in the chat. Let's see. Yeah, there's one question about caffeine, but it's not related to your presentation, Ronaldo. But maybe you know anything about caffeine. Can excessive caffeine early in the first trimester of gestation promote poor placental growth and induce early onset preeclampsia? Do you have any idea about that?
Well, we haven't tested the effect of caffeine, but as far as I know, there are reports showing that pregnant women that drink a lot of coffee show less symptoms and signs of preeclampsia. So it seems that caffeine is beneficial for the pregnant women with preeclampsia. And the same thing has been found with cigarette smoking. Apparently, there is a correlation, a positive correlation between cigarette smoking and a reduction of the symptoms and signs of preeclampsia. But we haven't tested this possibility.
Yeah, of course, we have to stress that the detrimental effects of smoking are weighing out.
Of course.
Well, the same
it's
is
not recommended at all.
No, the same is true for inflammatory bowel disease. I think smoking is helpful in colitis ulcerosa, and it's detrimental in Crohn's disease.
Yeah.
So
it's a similar finding.
I was just wondering about the chemical reactions of magnesium sulfate. If it reacts with O2, can it react with O2 minus or only with hydroxyl?
No. It's just with a radical. It's not with a functional group. It's the free radical, the hydroxyl radical.
And so, I mean, everybody focuses on the magnesium, but is there also a role in the beneficial effect based on sulfate? Well,
we have tried using different magnesium sulfates. Salt. Sorry, we have tried different magnesium salts, and it seems that the best magnesium salts are magnesium sulfate and magnesium gluconate. They are the best magnesium salts for the effect. So it doesn't seem that that the effect is related to the anion in particular, but to the particular magnesium salt. Is the whole thing the magnesium sulfate, or in the case of the magnesium gluconate, perhaps it's the presence of these anions that have around 10 groups OH. So
if you use other magnesium salts, you'd see the same effect?
No, it's not so beneficial. So when we try to use magnesium chloride or another magnesium citrate, it's not as effective as magnesium sulfate or magnesium gluconate. Those
Okay.
are the best. Those
Yeah.
are the best salts.
But in all these cases, the magnesium is the same molecule. So how do you explain the differences between the salts?
Well, perhaps the clue is that the magnesium sulfate does not dissociate like the magnesium chloride, because the magnesium chloride dissociates so easily into magnesium and chloride.
Yeah.
So perhaps that's the clue. But it's really intriguing that magnesium sulfate could be as effective. It has been used for almost 100 years. And it is the most effective therapy to stabilize a preeclamptic women with severe features. That is really amazing.
Okay, let me see. I don't see any more questions in the. Oh yeah, here. How did you define ghost red blood cells in your essays?
Well, ghost is jargon in the lab. The red blood cells are isolated, and then they are hemolyzed, and they are washed extensively by just centrifugation and resuspension, and we remove the hemoglobin, and what is left is just the plasma membrane of the red blood cell. And those plasma membranes or broken membranes of the red blood cells are white. And that term was suggested by Dr. Joseph Hoffman at the Yale University. He said, well, what is left is the ghost of what once was a red blood cell. And those are the red blood cells. Okay, and they are pretty useful preparation because they don't have any organelles, any intracellular organelles. It's just plain plasma membranes.
Yeah, thank you. And Hector says, thank you for the clear explanation.
Okay, thank you.
Thank you for the clear explanation.
I think that concludes our meeting for today. Ronaldo, Carmen, and Ricky, again, thank you very much for fascinating presentations. We enjoyed it very much. And for the attendees, thank you very much for asking all those questions, because they make those sessions very lively and interesting also for the speakers. I hope to see you all next week, and enjoy the day or enjoy the evening. Thank you again.
Okay.
Bye.